let's record the session and then we will upload it to YouTube when done. So go ahead, the time is yours.
Okay, great.
And just a reminder for the judges, you will still have some time to discuss all the things that you've seen here right after this session is done. So please go ahead, the floor is yours.
Okay, great. So great job, team. I think it's one of the more impressive mathematical simulations that I've seen. You know, I saw like a lot of the plots and graphs that you guys created, but if you were to see that when you're running this, it doesn't match up with what the models are predicting, what kind of things would you check to revise the model? What would be some of the first things that you would look for?
Okay, I can answer this. As you may have read in the Juggle, we base our model in a previous work from Team Chihuahua from 2017. They assume some of the variables. We checked some of these variables, but we also believe that some of them might need more investigation to confirm that these are in fact true. Well, this is some of our next steps that we plan to do to continue investigating and to even prove them inside the lab, especially the ones related to the enzymatic constants of both enzymes that we have been proving in the model.
Thank you.
Okay, judges, do you have any questions? Please go on, Sikak.
Okay, thank you very much. I just have I have one or two questions to ask the team. You worked on the development. You developed a biocontrol mechanism for Tijoko. Is it what you call it, Ewinia, that causes fire blight disease, which eventually leads to the death of the tree. Now, I want to ask, have you researched on, did you conduct any, or did you carry out any literature search, or did you research if there are competitors? And if yes, how would your projects be different from those of other competitors?
Who would like to answer that question?
Well, we already haven't searched about that, but I think there are no other like competitors that planned to do a biocontrol system like we are already planning to do. But in case there are some, we are looking for to see their work, and if their work it's like something that could help us to make our work better, we are going to use it.
Can I add something?
Yes, please.
As well, we researched before about if there were any other kind of mechanism that serve the same purpose. And we found that there are, but they doesn't really use the same mechanism that we are planning to use. So that is the difference, that basically we are using DMOs, and most of the other mechanisms are based on, I would say, practices that have been applied before and are traditional, we could say.
That's great. Thank you.
Yeah, I would like to add something more. Indeed, there's a lot of research related to the Fireblight and also to other QS systems. but there are not a lot focus indeed in the Tejocote. This will be like an innovation. Also, to help the fire blight, there are a lot of techniques that can be used, but there are, like my friend Aldair said, there are traditional ways that indeed are affecting, giving resistance to the bacteria, to Erwinia millivora. Therefore, this is like an innovation using the genetic modifications to help the problem.
Great answer. Thank you.
Okay, judges, please go, Beatriz.
Do you foresee any toxic effects from people eating the modified tejocote after your system is in place?
Well, since the biocontrol system is mainly, its purpose is to like kill the bacteria before the bacteria produces the infection in the plant, there will be, well, we think there will be no toxic effects in people when consuming the fruit, because the strategy is mainly, principally to attack the bacteria instead of making damage to the plant.
Great answer. Please go, Farouk.
Could I add something about this question? Okay.
Yes.
Regarding to the treatment, like about like the farmers are using with this kind of plague, I think that they are using a cupri substance, and I think that that kinds of treatment could rise some toxicology in the plant. So I think that our biosystems is better than they are using right now, right? Because we are using something that are not giving to the plant some resistance, like they are using.
Okay, thank you, Montserrat. Now please go, Farouk, with your question.
Okay, actually my question is about how you are going to implement your project, how you are going to administer your bacteriophage.
Do you have any ideas or thoughts about that?
Well, first of all, we, as I don't know if, well, I think you have already read on the Juggle that before we started all this research and all the project as a thing, we were previously directioned to talk with some people that work on this area, raising tejocote crops. And the first step on the application of this project is with them, with their crops, because in that place we found this bacteria. And so that would be the first step. And then we are planning on applying this on other fields or other states in the country.
Okay, Maria, you can add something.
Yeah, well, as Aldair said, we went in January of this year to see the agricultures and the people that work with the tejocote, and we make like an agreement with them that we're going to help them. But first of all, we want to make all the experiments in a laboratory, because if we go to the field, the bacteria could be spread and that would be catastrophic. So first we want to make the experiments in a laboratory with tissue culture of plants, and And yeah, first, that would be the first thing. And then if we see that our biocontrol system works well, we're going to go to field with these people that we already know.
Yeah. Also, as well as we know we can't do experimentation directly in the field, so we know that we have to do the experimentation into the laboratory. Yeah.
Okay. Yes, no, I'm just letting you know that we are already over the first 10 minutes. So please go with your question.
Okay. Are there any regulations in your government that contradict with your project? Safety regulations?
I can answer that question. No, in fact, we have not enough regulations. I can project something in my screen.
Yes.
Or, oh, it's okay. I can't. I need the authorization. Sorry.
Yeah, Daniel can help us with that. But meanwhile,
okay,
I
can go see. Okay. Yes. Well, in Mexico, we don't have enough regulation about biosecurity. So we promote to generate a new NOM project by the Law of Biosecurity of Genetically Modified Organism and General Health Law. Exists one project of NOM, but that doesn't include guidelines of biocontrol system process or evaluation of results and relevance. Also, we need to include and security control and a final disposal elements. For example, if we need to do something in the laboratory, we need to have the schedules for do that in a NOM. In Mexico, the normative framework functions in that way. So we need to check the international regulation that we include, like Cartagena biocontrol law, international law, and also the law of ecological balance. So we are in a step 1 in risk. So we need to develop a system in a NOM with Sagarpa, with Semarnat, that are Ministry of Environment and Agriculture. And also we need to work with local ministry in the state of Puebla, where is the Tejocote plant, because this can be a project that will be applied in a policy public in Puebla specifically. So I don't know if this is enough, because I wanted to,
it's totally fine.
to project.
Yeah, it's
okay.
Thank you. Thank you.
Thank you. That was a great answer. But judges, do you have any follow-up questions related? Well, I would like to ask you something. Was it a challenge to talk with all the farmers and the agricultors and producers and everyone? And how did they approach, and how did you approach your project for them to actually understand it? Because it's not easy to translate SynBio to the local communities.
Well, first of all, the jocote is the fruit, is one of the most important fruits in Mexico because of the impact it has by the food and also with the traditions that we have here. So we saw the need of the agricultures because their crops were infected and they were very affected by this bacteria. So the first step we wanted to cover was to go with them and also explain them first to explain them how the bacteria works in the crops, to make them know how it can be like killed, And we also gave them like, I don't know how to say it, but like talleres.
We also
yeah,
developed some workshops with
Workshops.
associations, and also we propose to continue doing workshops with Puebla local state or another ONGs, and also with the federal government in Mexico, because this can be applied in Tejocote in an initial state, that is Puebla, but also we can use it in other plants, but in a second or in a third step. But yes, we did a workshop. We have a model of workshop with an association, and also we want to, or we propose to make new workshops with the government and other associations.
Okay, Dimani, and after that, Montserrat, please with your final thoughts.
Yes, to continue with the first idea of Maria is that one of the first approach was to teach to the agricultors how bacteria works and some aseptic method to avoid that problem, like the use of alcohol in scissors, in some knives, and some tools that they use to cut the fruit. So it was very important that they know that to control in a slow level, because it's one of the main transmission of this bacteria.
Yeah, I think that one of the most important things before to do any kind of work, like how we could implement a biosystem, first of all is to learn about the farmers, because they are the people that is directly affected by this kind of plague or disease in the rosacea. So the mainly objective to the meeting with CESABEC, that CESABEC is an organization in Puebla, and we made an agreement with them that could give us a better approach in this kind of disease. So the first thing is obviously to learn about the farmers and how we could identify this kind of pest in the leaves or in the flowers, or I don't know. And with this kind of knowledge, we could implement a biosystem with sanitary biology.
Okay, thank you so much for your incredible answers. I don't know if you already have another doubt. No, because, okay, well, our time's up, and thank you so much for being all in here and for your incredible answers, and you did an amazing job with your project. So congratulations.
Thank you.
Congratulations, team.
Thank you.
Well done, guys.
Well, now I'm going to invite you to leave the session, please.
Thank you.
It was a
Have a
pleasure.
nice weekend.
Thank you.
Bye.
Okay, now we can have like a small discussion panel considering all the teams. If you have like a specific doubts related to the content of the teams, or I don't know how to do the evaluations or anything related to the judging.
Okay, actually, I have a question. You know that the rubric form, I thought it's something like I can edit it after submitting it, but it seems it's not like that. I submitted the first one, I think, with the presentation score. I had to select any score, so I made it 0 in case I can get back to it and adjust it. But I'm no longer can edit it. So I was just curious about.
Okay. Yeah, I'm going to let Daniel know so he can give you access again and also to check what we can do about that.
Okay, good.
I have a follow-up question on that, talking about deliverables. So what we submit tomorrow is the gradings. Do we have time to submit? Should the comments go on that? Like, can I give feedback and comments after tomorrow, or is tomorrow the deadline for everything?
I think no. I think feedback is not tomorrow, or is tomorrow.
Yeah, that was my question. Because on the working spreadsheet, there was a space for comments.
Okay, let me ask directly that because I don't know the answer about that.
Yeah, that is fine. Just looking to know how much time I have.
Yeah, don't worry.
I think the deadline of the feedback is on 13th of
Okay.
this month. I think, yeah, we have to make sure.
Yeah.
Yeah, because this is what I wrote when we had that presentation.
Same.
Yeah,
That was longer than the main competition. Yeah.
yeah.
I'm not so sure. I think that you can edit the comments later, but
Okay.
I'm just asking because something.
Yeah, better to be safe than sorry. Yes.
Yeah. After everything in here. So tell me, do you want to share something about the teams, about their presentations? If you have like doubts related also to the content?
I think the main thing that I missed from most teams was biocontainment strategies. And I was trying to gauge how much that was due to there's no regulation. So we don't have clear guidelines on what needs to be done. And how much was the teams not considering that, but I thought it was hard.
I think my consideration was too about biosafety and, you know, the biocontainment of the genetic modified organism, and how I was curious about how you are going to tell your system shut down in any moment. And this is what I was looking for each team actually, but I didn't manage to find.
No.
It's different. Yeah. Because in iGEM, there's stuff that you cannot even propose, right? Like a lot of the gene drives, no team here had a clear gene drive, but a couple wanted to modify the plant itself or the food crop itself, which I find non-ideal, but it's hard to, and I understand their side too, right? Like with no regulations being present, it's like,
yeah, that's the thing, because we're talking about Latin American
yeah.
countries, and I'm from Mexico. I know that issue, and in this case, we also need to work a lot in getting them to know that if you can't find a path, then your responsibility can be to actually build it, and if you are working with something, then think about all the possible scenarios and what would you do in each one of those scenarios, basically. But okay, I'm also taking that note so we can share it with the teams. And something else, something that you would like to share about a specific team, if you're not sure about the rubric, about their deliverables or something else.
So I saw that a couple of teams had, I'm talking about the information package for judges, that some teams are not eligible for certain prizes. So I'm assuming I just don't have to fill that session, right?
Yeah, no, in this case, the staff, well, the group they already know which one of the teams are related to that prize and which one didn't, because of the time issue, the deadlines and everything else.
Gotcha.
I don't know, Sikak, Rudy, do you have something else to share? Or Beatriz also?
No, I was gonna say, great job, Rudy, multitasking with their kids. That was impressive. That was very impressive.
I know, it was awesome. Yes, Sikak. I don't know if I'm pronouncing your name correctly. I'm really sorry.
Yeah, you pronounced you are pronouncing my name as if you are Nigerian. So I'm very happy to pronounce my name very well. So the teams, I observed that the teams were very conversant in their projects. They knew what they did, and they spoke as people that actually carried out their work, diligently. So I'm very impressed about their ability to defend their work.
Yes, I also noticed that they gave pretty good answers in a lot of times. But well, I don't know if you have any final thoughts, so I can let you all to rest for the weekend.
No, I'm just wondering, yeah, that time, what is the deadline tomorrow for? If I can enter comments later, that's my yeah. Oh, look,
here.
Daniel's here.
Yeah.
Let me answer that question. So the deadline is tomorrow midnight Boston time, New York
Okay.
time. So Eastern.
But is that for the, yeah, it's fine. Is that for the grading, or is that also for the comments and feedback?
No, that is just for the grading. So please relax with the feedback. You will have enough time to get them. Actually, we're going to give you, I think, 1 week.
Awesome.
And we will share with you another Robert weekend. Probably it was a bit long. With Robert for you, a bit long. So yeah, we split the feedback part. So you will receive one, and you don't have to worry right now.
Awesome, thank you.
Feedback for the teams, and you'll also receive a feedback for the judging process. So please also write down all your ideas and comments.
My idea is next year we clone Danielle as a project.
Please, you know,
Yes,
I almost had a heart attack today. I'm still in 2 separate rooms. I was streaming everything. I need at least 2 or 3 clones.
Yes, definitely. We need like 3. No, how many rooms do we have? We need like 5 Daniels, please.
Okay, Daniela, I have a question actually regarding the rubric form. I actually thought it's somehow editable, so I can get back to it and edit it. But yeah, unfortunately, the only one thing that I made mistake with is the presentation score thing. I made it for 0, and I told myself that I can get back to it and edit it at any time, but unfortunately, I didn't manage to get back to it. So I just.
If you did some error like this, just let me know, and I can manually correct it.
Okay.
So you can send me that break from that team, so I can check if it is wrong or right, and then we can manually change it.
Yeah, it's just one question, I think. Okay, thank you.
Okay, I think that if you don't have like any more questions, comments, thank you all for your amazing work, for your patience, and for being an amazing judge for the teams.
Thank you for the opportunity. Thanks, Sophia, for organizing.
Well,
thank you, Sophia. Thank you, everyone. And well, if you have any questions, you can write them in the Slack or via mail if you want, if you prefer. So thanks a lot.
Thank you.
Sure.
And see you on Thursday and Tuesday for the vote, the final vote.
Yeah.
Well, have a great day, a great weekend, night, evening.
Yeah, it's night. Okay, bye.